thing we observe is sex-biased expression. So sex-biased expression means if we compare the genes expressed on the X and Y chromosome, or just look at the gene expressed on the X chromosome compared to the autosomes. And we would like to know whether there's a particular expression pattern between the X chromosome and autosome, particularly in the sex-specific expression manner. So in Drosophila, the X chromosome is sort of like express more female biased genes. A lot of male biased genes are not exist on the X chromosome in Drosophila compared to the autosomes. So this is like if they are all randomly distributed, and you see the same amount or similar ratio of female and male biased gene expression. But in several Drosophila lineages, we observed female biased expression on the X chromosomes. And this also observed in mammals, but it's a little bit complicated. So in mammals, we observe female biased expression in placenta and also the ovaries. But we also observe male biased expression in the testes in terms of like just consider the entire X chromosome. So the pattern is similar, but it's slightly different. So this will explain later, because to explain the biased sex expression, we have to understand the X chromosome or XY chromosome were inactivated in many organisms during germline, and that is before the whole autosomal set were condensed. So the specific germline XY inactivation, and that could contribute to the sex-biased expression. From last lecture, we talked about the sex-biased expression, and this is only focused on the XY system. But this phenomena also observed in the ZW system. So in the ZW system in chickens, we observe female-biased expression is reduced. And in the silkworms, we also express the same phenomena, but I wrote it differently. It's the testis-specific genes are enriched on the silkworm's Z chromosome. So despite the different system, XY or ZW, it seems like the biased gene expression is also a common feature during the evolution of the sex chromosome. Also, like remind me, our previous lecture talked about the richer genes. So when we look at the biased gene expression, and I want to review like the idea we did with the relocation of the richer genes. When I say relocation, actually remember the study from Wang Yuanlong's group. They compared the richer gene and their ancestral copy and illustrate where is their ancestral copy located. It's either on the X chromosome or on the autosome. Then they also point out the position, the chromosomal position of the richer genes. And now you can look at the distribution of the ancestral copy and the richer gene, and there's a biased distribution on that. So this is also has something to do with the biased gene expression, and the mechanism, the underlying mechanism, we think one is the chromosome, particularly like an X chromosome, they stay in the female for much longer time than in the male. So there are 2 X chromosomes in females, but there's only 1 X chromosome in males. So if there's any female beneficial mutations on the X chromosome, it will be easily fixed in the population. So, or even more, some sexual antagonistic allele, that means the allele is only beneficial for one sex, but deleterious to the other sex. So the distribution of this type of allele will be also different between the two sex chromosomes. The other thing is what I mentioned in the previous slide is the early XY inactivation in the germline. So that means if a gene is essential for spermatogenesis, and on the X chromosome, you have to ensure the expression of those genes has to be turned on or complete before the X inactivation. If anything happened after, or there will be very few expression of the X-linked genes after the inactivation events. So this probably all contribute to the biased gene expression. So this is like what I put here. And so the selection on the sex-linked partially dominant mutation, which beneficial for the homozygotic sex. So now I didn't say like a female like us. So homozygotic sex like female in mammals and the males in chicken and butterflies. And also there's a meiotic sex chromosome inactivation. Some like a lot of review paper will just write MSCI, and that probably also contribute to the biased gene expression. Another thing we found recently with the genomic sequence is on the X and Z linked structure. So when we say the X and Z linked, it's like the X chromosome and the Z chromosome share the very similar genomic structure. That is, there's a replication or duplication of particular locus or loci on either the X chromosome or the Z chromosome. And this is a very sort of like a special gene structure. Like we call it epiconic gene structures. So to illustrate this, I want to show it in this slide. It's the illustration from the Z chromosome. And the epiconic gene structure is showing on the right of this slide. There's a relocation of one particular locus, and after this relocation, there's amplification of this locus indicated by several errors in a string. So this replication results in, so there's also results to the amplification of the entire region. So they are repeated sequence on the X chromosome. So this was illustrated, so one replicon showing here. So there are two replicons, and repliconic region is showing here. One is for a single locus, they replicate several times, and the other one is amplicon contains 3 different genes and also duplicate multiple times and on this chromosome. So this is sort of like a special gene structure on the Z chromosome here, but we also observe on the X chromosome as well. So this seems like compared to the autosomal regions, this is a very special feature of the X and Z chromosome.